Today we will perform ZN stain, means Ziehl Neelsen stain, and it is also called AFB stain, means acid fast bacilli. This stain is important to isolate or diagnosis of Mycobacterium tuberculosis. Before ZN staining, we have to prepare a smear. For smear preparation, we need sample, sputum or any fluid, glass slide, nichrome loop, and also Bunsen burner or spirit lamp. Before smear preparation, it is very careful that we have to need safety precaution, because acid fast bacilli is too much aerosol in nature, so it can easily contaminate with air and infect any human being. Preparation, we have to need one tissue paper and we make a smear over that paper. So after processing, we can easily discard. So it is the most important part. Okay, look very carefully what are the step. This is a spirit lamp. This is the nichrome. Okay, what will happen at first? We burn up the spirit lamp, and we should remember that at the time of smear preparation, always the smear will be prepared in back side of the lamp, means this side. Okay, so what is the reason? Reason is that at the time of smear preparation, the aerosolic nature of Mycobacterium tuberculosis, it never be infected to close person. So it's create a heat barrier line. So it also protect to contamination. You have to be very careful about that. At the time of smear preparation, we need to wear gloves, face masks. N95 mask is very efficient, very useful, and if you need, always wear head mask also. So at first we have to sterilize the nichrome air loop. This is the ready procedure. After that, cooling it properly. Then we open the container with supplied sample. This is sputum sample. After that, we mix it properly with a nichrome air loop and make an take little amount of sputum sample and cover it carefully. And always remember that the smear will be prepared backside the flame. So like this. Okay. That time we also heat fixed it. After preparation of smear, heat fixed it gently. A reason of heat fixed is the one is the decontamination, another one is the mucoid part of the sputum or any other clinical sample. It get easily attached with the glass slide. So at the time of washing, it never wash off from the glass slide. After that, we again sterilize the micromer loop. After preparation of smear, what it is very important step, we have to clean the area properly. And first, this is the hypo solution. We have sprayed the hypo over it. 1% sodium hypo over it and fold it up very carefully. After that, the area should be cleaned up properly with cotton. After cleaning with 1% sodium hypo, we have to switch on the UV light in a laminar airflow, and we should remember that the total process should be completed within closed laminar airflow. Now we will start staining procedure. First one is we put a carbolfuxin over the smear and take a cotton and heat it gently. Okay. Similarly, we can take a test tube with carbolfuxin and heat it gently, and should be remember when vapor will be arise from the test tube, that time we can put over the smear. and allow it for after holding 5 minutes. Discard the carboxyl solution and wash it properly with tap water. Then put 20% H2SO4 for decolorization. And we have to notice that until decolorization, we have to observe the smear. Discard the 20% H2SO4 and wash it properly with tap water. After that, we put methylene blue as a counter stain and allow to hold up to 30 seconds. Discard the methylene blue, it properly with tap water. After washing, then allow to air dry. Complete air dry procedure. Now we seen it. The smear will take the bluish in color, the counter stain of methylene blue. Now we are observing it for microscopy. For observation of the smear in microscopy, we have to put immersion oil. Without immersion oil, it never be seen in 100x or oil immersion objective. Seen, the slide is placed under the microscope and we have to observe under the 100x or oil immersion objective. Now go for the microscopic observation.